We are going to talk about the stereochemistry of vitamin, or specifically only vitamin A, which is retinal. It's an aldehyde. It has conjugated double single bonds. Carbon 11 is important because you could have a cis configuration or isomer of this retinal versus a trans isomer of that retinal. So this is known as isomerization, and light energy can do that. Now, visual cycle is a really transformation or conversion of light into electrical signals. Back of our eye, the retina contains two photoelectric sensors or cells. One is cone shaped, the other one is rod. And the rod contains a protein known as opsin, which can attach to the cis 11 retinal and transform into rhodopsin, another protein, and this is when the visual cycle or electrical signals are going to bring. So to look at it, and few things to remember is what happens first is the opsin, the protein in the rods in back of the retinal, and the cis retinal, your vitamin A, are going to attach to each other, but it's a reversible bind, meaning it can go back. It forms rhodopsin. Then in presence of light, this rhodopsin and this rhodopsin is going to convert the cis into trans, but the trans is not a perfect fit for the protein, and there will be a change in configuration of that protein and causes an electrical impulse going to the brain. This triggers a response, and then you can visualize something and see something. Now what happens at very end, due to enzymes, the trans is going to convert back to cis, and it separates from opsin, and this entire visual cycle sort of takes place again and again. And this is what your very last stereochemistry is all about vitamin A and its two versions of cis and trans, and how it's going to sort of create signal of visual signals. And you have to remember the processes, and you have to remember sort of the process that happens 1, 2, 3, and finally the separation.